Good morning. The subject that we want to discuss this morning is related to the role of the immune system in reproduction, and is an understanding of what the immune system is doing and the interaction between the mother and the fetus, especially the maternal immune system. When we look at the pictures like this one, that is a classical case of a baby born with a disease that is called the hemolytic disease, where the case is that the maternal immune system is attacking the red cells of the baby and destroying the baby's cells. Or when we look at complications such as recurrent abortions or miscarriages, where the maternal immune system really is rejecting the implanting baby, the question that always comes to this is, what did your mother reject you? Yeah? What is happening that the immune system of the mother is not willing to accept the implanting baby? Pregnancy, in reality, is a very unique immunological condition. Why? Because, as we discussed during the next hour, the immunology of pregnancy has this paradigm that requires a maternal tolerance to paternal antigens that are present in the fetus, as well as at the same time preventing or protecting the fetus against infections that comes from the environment. The concept of an immunological tolerance was introduced more than 50 years ago by an immunologist, a transplant immunologist, Sir Medawar. And Sir Medawar recognized the paradox that the fetus, in genetic terms, is a semi-allograft that escapes rejection. What it means that the baby, it's made of 50% of paternal antigens, and those paternal antigens, when it's implanted, are recognized by the maternal immune system. Very similar is we take a piece of skin from the father and we put it in the mother. The maternal immune system will recognize as foreign body and immediately will reject it. So the baby, in many ways, is like a piece of skin from the father, although has some components from the mother, but is recognized by the maternal immune system, and in majority of these cases escapes this rejection, and that is the reason we have successful pregnancy. So Medawar, when was working in this concept of transplantation, he proposed that a natural example of transplantation is pregnancy. And in his hypothesis, the fetus, a semi-allograft, as I indicated before, escapes rejection through mechanisms that involves a systemic immune suppression. So similar as today, when we do a transplant of an organ like a kidney into a patient, in order that kidney will be accepted by the host, you need to immune suppress the organ. So he proposed that in nature, there is a mechanism, a natural mechanism of immune suppression that has allowed the success of the pregnancy. And since these observations, the area of immunology of pregnancy has focused on this concept of graft-host response. So we're going to evaluate some of the concepts trying to explain, or the mechanism trying to explain this graft-host response during pregnancy, And we will evaluate whether this concept is true or not after almost 50 years of investigation in this area. The immune system in pregnancy, as I mentioned, is unique. And there is an aspect that is very important, especially in these days associated with pandemics, such as the COVID-19, because pregnant woman has been always considered a high-risk population. Why? Because they develop severe complications, such as mortality, preterm labor, in times of viral infections, in times of pandemics. And one of the explanations why pregnant women represent a major risk population is with this idea that they are immune suppressed. So when you have a patient or you have an individual that is immune suppressed, his or her defenses against the viruses or bacteria are decreased, and consistently they are in the risk of being affected by the pathogens. And in addition, many of the pregnancy complications, as I mentioned, such as recurrent abortions, miscarriages, and so on, as well as even preeclampsia, which is a complication that develops much later in the pregnancy, all of them have the question whether this abnormal pregnancies is due to the lack of an appropriate immune suppression. So let me review very quickly, for those who are not familiar with the early stages of pregnancy, what are the components that are going to be interacting with the maternal immune system. And what I'm showing you here is the embryo at the implantation site. Yeah. What we see here is the blastocyst penetrating within the uterus. And once the blastocyst penetrates within the uterus, will interact with the cells what are the uterus, and we'll talk in a minute about that. The embryo can be divided in two groups. One is the inner cell mass, that we see here, and the second one that gives origin to the fetus, and the second one that is the external layer that is called the trophoblast. And the trophoblast gives origin to the placenta. And why I bring this, because there is an interesting phenomenon. The placenta, that is the organ that will be in direct contact with the maternal immune cells, either present in the uterus or the maternal immune cells present in the circulation, is made only or mostly of paternal origin. So 90% of the proteins that are expressed in the placenta are from paternal origin and not from maternal origin. So that suggests that the cells who are going to be interacting with all the maternal immune cells are really completely different and foreign to the host, in this case the mother, making it even more difficult to understand why those cells, the placenta, is not rejected by the maternal immune system. So if we put it in a nice and simplistic way, that beautiful baby that you see there is a combination of the mother genes and the paternal genes. And in such a beautiful baby, the mother never will reject the baby. On the other hand, the placenta is mainly paternal. And it's not surprising that in many cases, the mother will like to reject some things coming through the father. So based on this, several years ago, we tried to redefine what we call the immunology of pregnancy. Because in reality, it's not a fetal-maternal interaction. It's really the trophoblast cells that are interacting with the maternal with immune cells in the maternal circulation, or the same trophoblast cells who are invading the uterus are interacting with immune cells present in the uterus. And we will talk about that in a minute. So in reality, when we talk about in pregnancy, the immunology of pregnancy is not maternal fetal immunity, is really maternal trophoblast immunity. Why we call pregnancy a unique immunological condition? Because if we take a sample of the uterus at the time of implantation, and we look at the cells that are present in the uterus, we'll be surprised that the most dominant cell type, the cellular component of the uterus, are immune cells. And when we look carefully about what type of immune cells are present at the implantation site, we see that you have NK cells, T cells, dendritic cells, CD8 positive cells, macrophages, and so on. NK cells, which are the big attackers or rejectors of foreign bodies, represent 70% of the cells that are present in the uterus. The presence of these immune cells at the implantation site was considered in the early stages of when Sir Medawar was proposing the idea of a graft, was the proof that there was a maternal immune response to the implanting fetus. And Medawar proposed that when the fetus implant, there was a migration, an input of maternal immune cells that they really were trying to attack that foreign body that was penetrating within the maternal tissue, that is the uterus. So what are the types of immune cells that are present at the implantation site? So although you are experts in immunology, so I don't need to go in all the details. So we know that we have two arms. One is the innate and the adaptive immune responses. In terms of the innate immune system, what I mentioned before, you have the macrophage, the granulocytes, the NK cells, and the dendritic cells. Their role is to patrol and phagocytize foreign materials. All those innate immune cells are present at the implantation site. And when we look at the adaptive immune response, talking about the cellular, the cytolytic response by T cells, or the humoral response by B cells, we found that cytolytic T cells are not present or are in very low number present at the implantation site. There are some cytolytic T cells, the CD8 positive T cells, very few. And it's interesting also in terms of the humoral, the B cells producing antibodies, there is also very little of B cells. However, you have a lot of T regulatory cells present at the implantation site. So looking now at these two types, the T helper cells, if we divide them in the Th1 by interferon gamma, that increase cell-mediated immune response, or the Th2, that is mainly anti-inflammatory, the production of interleukin-4, interleukin-10, and so on, they increase the humoral immune responses. So those different types of cytokine environment, we will discuss also later, both of them are present at the implantation site, although at different times. Now, in general, when we call about the cytokine environment, just as a quick introduction, we will develop a little more, we call the Th1 as the pro-inflammatory and the Th2 and the anti-inflammatory. And it has been suggested that the non-pregnant woman, in general, is a pro-inflammatory, and during the pregnancy, there is a shift in a systemic way, and it becomes an anti-inflammatory condition. So that again, all the different arms that you became familiarized in this course about the immune system, they are present specifically within the uterus of the mother at the time of the pregnancy and the implantation. So the next question is, what is the terms of the allograft, how it responds to the maternal immune system. So very quickly, just to remind you, what is the different types of transplant immunology. If you take a piece of skin from yourself and we put it in another part of your body, that is called an autograft and is not rejected. However, if we take a piece of skin from, let's say again, from the father and we put it in the mother, that is two different individuals, it's an allograft and there is a rejection. Or the third type of transplants is you take from a different species and you transfer to another species. That is a xenograft and is rejection. Pregnancy, as I mentioned to you before, it has been considered in the middle, a semi-allograft. And in principle, it should be rejected. So what we have been trying to understand is why, since this is an allograft or semi-allograft, why is not rejected. And one of the early experiments in this area was done, again, many years ago, and using this type of animals experiment. So they took just, let's say, a white mice and took a piece of skin from a black mice and transplanted into the white mice. That skin was rejected. But when they crossed these two different strains, and they did the same experiment, transplanting a piece of skin from the father, in this case the black mice, put it in the mother, who is the white mouse and now is pregnant, now that piece of skin was not rejected. So this is the background understanding what is all about, or the different challenges, immunological challenges that is happening during the pregnancy. So now the question is, how do we explain that? And of course, there are several hypotheses trying to explain why there is no a rejection. And I will go through the main ones, and I will present you at the end what are the new explanations of the new data that we have. One of the early explanations why the fetus was not rejected is the existence of a mechanical barrier. The second one is the suppression of the maternal immune systems, and what I will discuss now is also the Th2 type immune response, who are relevant for what is going on. So let's start with the mechanical barrier. In our body is thought to exist several organs who are isolated from the rest of the body. For example, the brain, the eyes, the ovaries, the testicles, and the pregnancy. For many years, we thought that those organs, also known as immune-privileged organs, are isolated from the immune system by mechanical barriers. Like in the brain, you have the blood-brain barrier, which will not allow the movement of immune cells or microorganisms from the body into the brain. In pregnancy, the barrier was thought to be the placenta. And the placenta was considered as a wall, as a bag that is contained within the fetus, and nothing from the fetus will go out, and neither immune cells from the mother will go into the fetus. This seems to be a wrong hypothesis, because now we know that all these immunological privileged organs really are not isolated because of the presence of mechanical barriers, but because there is very nicely controlled immunological mechanism that again controls the movement of immune cells in or out of those organs. In terms of the pregnancy, the placenta is not a mechanical barrier, because fetal cells are migrating into the maternal body, as well as maternal immune cells are migrating into the fetus. So we know that, for example, T regulatory cells from the mother are going to the fetus, and they have an important role on educating the fetal immune system. On the other hand, also, cells from the fetus are present in the maternal circulation. And here is an interesting aspect, because these fetal cells are present in the body of the mother, not just at the time of the pregnancy. They will remain for the rest of the life of the mother. What do I mean? That the fetal cells, who are stem cells, are all the time migrating during the pregnancy into the maternal circulation, and they will incorporate into the maternal tissues. And once they incorporate into the maternal tissue, such as the liver, the heart, kidney, and so on, they will differentiate into those organ cells, and they play an important role on tissue repair. Again, there are examples like in the liver of fetal cells that go to the mammary glands and prevent breast cancer and so on. In other words, every pregnant woman becomes a chimeric. That it means that woman will be carrying the paternal genes that were transferred by the fetus for the rest of her life. And I always like to say that based on these learnings, especially for those young ladies who are not married, this is the important message. Choose carefully your husbands, because you will be carrying his DNA for the rest of your lives. So there is no barriers preventing the movement of immune cells. So we cannot explain the lack of rejection due to this barrier. So it needs to be a more specific mechanism, because the exposure of paternal antigens is going to be for the rest of the life. The second hypothesis that has been predominant in the field, and even today many people still talk about, is suppression of the maternal immune system. So it's a concept that is everywhere in publications, books, and so on, that pregnancy is characterized by a state of immune suppression. Similar as the graft, the maternal immune system is suppressed during pregnancy, and that is what allows the fetal growth. And again, as I say, this is a concept that has been dominant for many, many years. Now, we evaluated and thought a little about this immune suppression and evolution. And if you look at the world, immune suppression during pregnancy doesn't make much sense. Why? Because if you think that the majority of pregnant women in the world, 100 years ago or 1,000 years ago, the pregnant woman were working in the fields, were continuously exposed to a not a clean environment like is today for majority of the pregnant women in the developed world. Those women, as I say, they're working in the fields, they don't wash their hands, they eat and so on. And I have this picture, for example, there is a painting from the 1600s showing a pregnant woman in the fields. If that pregnant woman will be immune suppressed, the microorganisms present in the fields or in the environment will have killed her immediately. And it will kill her if the pregnant woman, immune suppressed, is exposed to the environment and is weak, the species will disappear. Is this pregnant woman immune suppressed? As you see again, this is a woman that is happily pregnant, working in an area that it doesn't have the sterile conditions that we are characterized. Yeah? She doesn't die. The fetus doesn't die. She has a successful pregnancy. And we have another evidence that during pregnancy, women are not more susceptible to infections such as HIV. HIV, when a pregnant woman gets infected with HIV, or if an HIV woman gets pregnant, yeah, she doesn't suffer from AIDS-like disease. The opposite, during that time, the immune system is nicely controlled. Yeah. So clearly, this uniqueness of the immunological conditions of pregnancy is that during pregnancy, and again, this is an important concept that I want to leave you. during pregnancy, the maternal immune system is not suppressed. The opposite is stronger than ever. And we are seeing today pregnant women infected with COVID-19, they are not dying more than the non-pregnant woman. The opposite, they are doing very well. Why? Because their immune system is capable of controlling the infection. Yeah? And in addition, systemic immune suppression during pregnancy represents a danger to the species. Yeah? Because the most important evolutionary period in the human species or any species is the pregnancy time, is when the species is maintained, the genes are transferred. And if during that time the female is immunological compromised, the species will disappear. So nature has created a strong mechanism or unique mechanism that make the pregnant female, animals or humans, is strong to resist the environment. But that still doesn't antagonize or is against the concept that pregnancy is a semi-allograft. So those paternal antigens are there, and the immune system is recognizing those paternal antigens. So still we don't have the answer why they are not rejected. But clearly it's not because there is immune suppression. It doesn't exist a systemic immune suppression. So let's go back to the immune cells. So I said the maternal immune system is not suppressed. But we know that there is a lot of immune cells present at the implantation site. So are they attacking the fetus? Are they recognizing the paternal antigens and trying to get rid of them? So to answer that questions, we and others, what have done is using mice models to deplete the immune cells present at the implantation site, thinking that in that way we can help the tolerance. And what we found is that depletion of immune cells at the paternal interface, instead of helping the pregnancy, has a detrimental effect on the survival of the fetus. Yeah. So in summary of many years of work and information, all those immune cells that are present at the implantation site, they are there not because they are attacking the fetus or the placenta. They are there because they are necessary for many of the aspects related to pregnancy. They are important for implantation, they are important for trophoblast invasion, as well as for parturition. So, all what I have shown you is really that the concept of pregnancy as a graft host immune response is incorrect. And although this concept proposed by Sir Medawar and give him the Nobel Prize for this observation, after more than 50 years of this Nobel Prize, we can say that it was wrong. It was right with the observation that pregnancy is a semi-allograft that is made of paternal antigens. It was wrong with the explanation that is a classical graft. In reality, pregnancy represents a different model of immunological balance or immunological recognition. And we proposed several years ago that pregnancy may be more as a tumor host immune response rather than a graft host immune response. What I mean as a tumor? Let me compare the two conditions. In the graft host immunology, if we take the kidney and we put it in a host, in the moment that we implant that organ, there will be a massive release of antigen because this is a big organ. And in addition, there is immune cells, especially dendritic cells within that organ. and they will migrate into the host. And when they migrate into the host, together with a high amount of antigens, they will present to the T cells and the lymph nodes, and they will do what is called the classical immune activation. Expansion of effector T cells, activation of cytolytic T cells, recognition of those antigens, and then it will go and lead to the rejection of the transplant. The tumor, on the other hand, never started in the size of a kidney. The tumor starts with a very few cells. And as the tumor progresses, it starts secreting or releasing what we call the tumor antigens. And those tumor antigens are recognized by dendritic cells of the host. When the dendritic cells recognize a small amount of antigens, they take those antigens, they go to the same lymph nodes, but because of the quantity is small, and those are host dendritic cells, they will present to the T cells. And again, because of the quantity, as you know very well, it will induce the deletion of those effector T cells, and that is a process of tolerance. And we know that tumors induce tolerance. That is the reason they escape immune surveillance and progress to develop big tumors. Pregnancy never starts like a kidney. I never have seen a woman who suddenly appears with a fetus who is 9 months. Pregnancy, as you know, starts with 2 cells, then 4, 8, 12, morula, blastula, and so on, increasing the number of cells. As the number of cells increase, there is the leak of antigens, including the paternal antigens. And exactly as the tumor, the maternal dendritic cells who are present in the uterus capture those few antigens, it takes them to the maternal lymph nodes and start doing exactly like in the tumor, induce the deletion of effector T cells and induce tolerance. And this tolerance is forever. And that explains also why, even after the pregnancy, those fetal cells present in the mother will never be rejected, because it was established a very specific deletion of T cells that recognize the paternal antigens. So that is about the antigen presentation. So in reality, a pregnancy is like a tumor. Release of a small amount of antigens that leads to the induction of deletion of immune cells. Now, what about the immune cells? I mentioned that there was a lot of immune cells present at the implantation site. And we know also that tumors are characterized by the presence of immune cells within the tumor. And we know today that those immune cells present in the tumor have somehow transformed, and instead of rejecting the tumor, what they are doing is they are helping the tumor to grow. So that suggests that is an immune modulation. There are factors secreted by the tumor that are regulating the function of the immune cells, including macrophages, conversion of T regulatory cells, and there are factors, for example, the PD-L1 that binds to PD-1 and T cells and also induce the deletion, the cell death of those T cells. So the tumor is actively secreting factors that are preventing a rejection. The same is happening during pregnancy. The placenta is able to secrete factors that will transform those maternal immune cells into pregnancy-supporting immune cells. So the NK cells, for example, who are natively rejector cells, they are cytolytic cells. The NK cells present in the uterus are transformed by the factors secreted by the placenta and become NK cell regulatory cells. And they are preventing inflammation, but at the same time, they are protecting against infections. And we will talk about infections very quickly at the end. So, to summarize what I'm telling you, pregnancy is characterized by the existence of a specific cellular and molecular mechanism that tightly regulate the immune landscape without compromising the tolerance. So we have covered already the aspect of antigen presentation, the graft. We have covered the immune cells. They are modulated and changed. So the third aspect that we talked at the beginning was about the environment, the inflammatory environment. Pregnancy has been defined as an anti-inflammatory condition. And when we talk about anti-inflammatory condition, this is very important because when a pregnant woman has detected a condition of inflammation, immediately she was treated with anti-inflammatory conditions, with anti-inflammatory drugs in order to prevent the damage of the pregnancy. Yeah. The aspect also of this Th1, Th2 hypothesis, the anti-inflammatory condition of pregnancy, if we look at the literature, we see that there is a lot of contradictory data. Some studies show that indeed pregnancy is anti-inflammatory, but there is another report that will say the opposite. We did a study and we look comparing the first trimester of pregnancy versus non-pregnant in the blood, looking at the cytokines. And the arrow is showing you some inflammatory cytokines. And we look at the non-pregnant versus the first trimester of the pregnancy, and we saw that was the opposite. The inflammatory cytokines were going up in the first trimester of the pregnancy compared to the non-pregnant. So there was inflammation in the first trimester. We compare then what is going on in the different trimester of the pregnancy, the first trimester, second, and third. And here came the surprise that in the first trimester of the pregnancy there was a lot of inflammatory cytokines, and they went down in the second trimester, but they increased again in the third trimester. And that's when we propose that pregnancy may be divided in at least 3 environmental or immunological conditions in terms of the cytokines. A first trimester that is inflammatory condition. Why? We know that pregnant women in the first trimester, they feel terrible. Many of those symptoms were attributed to the hormonal changes. But the major hormonal changes come later. But if you look at those symptoms, are many associated with symptoms that you will see in patients with an infection. And when we look at some classical infection-related cytokines, such as TNF-alpha or IL-6, those cytokines are highly expressed in the first trimester of the pregnancy. And we will talk about why that inflammation is necessary at the implantation site in the first trimester. The second trimester really is the stage of anti-inflammation, or is the anti-inflammatory, or the Th2. And we know that at that time, the woman feels much, much better. It's the best time of the pregnancy. The third trimester, or the time when it's getting closer to the delivery, that's when it comes, again, the inflammation. Why? Because inflammation is required for parturition. The inflammatory process is necessary for this rejection, that is parturition. Parturition is taking out that foreign body. If we inhibit inflammation at the time of parturition, there is not, we block the parturition. So inflammation is required to parturition. And again, 2 stages of inflammation, early pregnancy and late pregnancy. It's just in the middle of the pregnancy where we can see this anti-inflammatory condition. So to put it in a graphic, we have, there is an anti-inflammatory before the pregnancy, at the time of implantation in the early pregnancy is a very strong pro-inflammatory, and then there is a switch from pro-inflammatory into anti-inflammatory. That anti-inflammatory is much of the second trimester or much of the whole pregnancy. And then there is another change, another shift in the environment, in the body, that leads to the inflammation necessary to parturition. So a major question that we're trying to understand today is really what regulates those shifts from the anti-inflammatory into the pro-inflammatory and vice versa, from the pro-inflammatory into the anti-inflammatory. Now, why pregnancy is, why do we need inflammation during the time of implantation? Again, we can take a similarity to the tumor. In this case, when we compare with tumor immunology, we are referring to the case of metastases. When we look at the metastases, that means cancer cells migrating into another site and implanted in another tissue. When the metastatic cancer cells implant into a new tissue, they attach and they invade. And all this process of attachment and invasion and proliferation within the new tissue really is due to the triggering of a classical inflammatory response. There is an inflammation that is allowing those cancer cells to attach to the tissue, and there is this inflammation that is helping the cancer cells to migrate within the tissue, and also to create new blood vessels, to heal the tissue, and enhance the repair. And when they enhance the repair, that repair is associated with the proliferation, not only of the normal cells, but also the proliferation of the cancer cells. That is what is happening in pregnancy. There is an inflammatory process in a specific area of the uterus that is going to change the surface of the uterus, allow the blastocyst to attach, or the trophoblast to attach, invade, and the inflammation that is happening within the tissue allows the formation of new blood vessels, helps the trophoblast to continue invading within the uterus, and will send signals to that blastocyst to start growing and the cells to proliferate. We know that because in animal studies, we have shown, for example, what I'm showing you here is the blastocyst implanting inside of the uterus. When we remove immune cells like dendritic cells, antigen-presenting cells that are extremely important in triggering an immune response. In this case, there's dendritic cells in the early pregnancy. They are responsible of changing the inflammatory milieu, and those dendritic cells act in the surface epithelium of the uterus and help the blastocyst to implant. If the dendritic cells are depleted, the blastocyst is not able to attach, as you see in this picture. It struggles with the epithelium, and at the end it will fail and the pregnancy will not happen. Again, an example that an immune cell is triggering an environment, an immunological environment characterized by inflammation that is critical for adapting the cells in the uterus to receive the fetus. So to put in summary what we know today, the dendritic cells secrete inflammatory cytokines and chemokines that will remove a layer of mucins that are present in the surface epithelium of the uterus, will induce the expression of addition molecules that will allow the attachment of the embryo to the epithelium, and then additional signals will tell the trophoblast to start invading, and that is what secures the implantation. If this doesn't happen, if we inhibit the inflammation, or if there are factors, we will prevent the migration of the dendritic cells, there will not be implantation. And that's what we see in many women who have implantation failure. The same as for many other diseases, timing is everything. If the inflammation occurs too early, we have a parturition at the wrong time, that is called preterm birth. If the inflammation never changed into an anti-inflammatory condition, and we have an inflammation that is too early in the first trimester, that is the miscarriage. And if there is no inflammation, as I mentioned to you before, there will not be implantation. So again, the immunological milieu, the immune cells, are extremely important for the normal biological process of reproduction. So STEM, what I mentioned to you before, what we don't know is what are the factors that are controlling these changes from pro-inflammatory into anti-inflammatory. So the last part of what I want to show you is the aspect, and again, we are testing this hypothesis that are factors who really convert the pro-inflammatory condition, factors secreted by the trophoblast that convert this pro-inflammatory into an anti-inflammatory. So the last part that I want to cover in this lecture is the concept of the microorganism. For many years, we have considered the uterus, and the pregnant uterus especially, as in a sterile environment, that there is no microorganism and there is no either signals from microorganisms reaching the uterus. Any time where we will see bacteria associated with pregnancy will immediately give us an alarm, because that we will consider we correlate with infections. And we know that bacteria exist in the vagina. And the bacteria could migrate from the vagina into the uterus, reach the placenta, and sometimes even reach the fetus. But every time when the bacteria will reach the placenta or the membranes, we will see that there is a disease. For example, if bacteria goes to the membranes, there is a complication that is chorioammonitis. Or if it goes to the placenta, it's called placentitis, as well as velitis. So those are inflammations of the placenta. And it will go to the fetus, it will induce, as I say, the fetal death. That has been the concept for many years. However, multiple studies are showing that the presence of bacteria alone is not sufficient to cause inflammation, fetal death, or preterm labor. Moreover, the use of antibiotics did not reduce the rate of preterm birth. And we can find, as I will mention to you, intrauterine infections that are in the absence of any type of bacteria. Yeah. Especially the aspect that the use of antibiotics did not reduce the rate of preterm birth, the consideration that it's not only the bacteria what is producing the problem, and that there must be bacterial signals that are present there. And there is now data showing that in the non-pregnant female reproductive tract, there is a normal microbiome. The aspect whether the placenta has bacteria or not is another subject. We don't think that within the placenta there is bacteria, because bacteria is not present within any tissue. It's always in the surface of the tissues. So we did an experiment that I want to share with you a few years ago. That is where we took E. coli, and we labeled that E. coli with the green fluorescence, GFP. We delivered that bacteria in the vagina of the mouse, and then we monitored what happened. So again, this is 12 hours after we put the bacteria, and you see that the fluorescence is in the vagina. And that is not surprising. That is the place where we put the bacteria. So it's concentrated in the vagina, and as I mentioned to you before, the vagina, we know that the vagina has a lot of bacteria. But the surprise was this, that within 24 hours, that fluorescent is spread through the whole uterus, the two sides in the mouse. And not only that it goes through the whole uterus, it's also reaching the ovaries. That is in the top. And in 48 hours, the signal is very strong. So the continuous migration of bacteria from the lower reproductive tract all the way up. The interesting thing is that 72 hours, the signal starts going down. And again, 4 days, 6 days, the signal almost disappeared, the signal of the fluorescence. However, if we look at the presence of GFP using PCR, we see that the GFP is always present there, even 2 weeks after we deliver the bacteria. In other words, two aspects that we conclude from here. One is that the bacteria is able to move from the lower reproductive tract all the way through the uterus to the ovaries. Second, and this is happening all the time, even in the normal bacteria. So the uterus is not sterile. All the time you have the movement of commensal bacteria. But the second aspect is that the uterus has an active and a strong, or has developed active, strong mechanism to control that bacteria and to reduce the amount of bacteria, as I show you in the reduction of the signal within 72 hours. And when it controls, the number of bacteria remains a minimum. So the uterus is an active mucosal, has a very strong mucosal immunity, capable of regulating the amount of bacteria present in the uterus. So this is really a summary of what we know today in terms of the immunological and bacterial condition in the uterus. We have all the immune cells that are known, they are present at the implantation site, as indicated to you. And in addition, in the lumen of the uterus, in the surface of the epithelium of the uterus, you have bacterium that is attached. And this bacterium is secreting factors or releasing factors that are recognized by the placenta, by the trophoblast, and are responsible of helping to modulate or to keep the maternal immune system at the implantation site awake when there is infections coming from the maternal circulation or from the reproductive tract, such as viruses or pathogenic bacteria. If we deplete this normal commensal bacterium, that awareness of the immune system is affected, and that is when there is risk for infections. And the immune system who was there protecting, it will shift when there is an infection, and will then, at the first stage, will battle the infection, but at the same time, we'll have to deal whether maintaining the pregnancy or terminating the pregnancy in order to protect the two sides, the maternal and the fetal side. So this is the contradiction of the pregnancy and the importance of the balance, the immunological balance on the pregnancy. Because at the same time, when these immune cells are protecting and helping the growth of the pregnancy, if bad signals are coming, the same immune cells will change and now will induce the rejection of the pregnancy. So pregnancy is a unique immunological microbial condition. The maternal immune system is not suppressed during pregnancy. The maternal immune cells are critical for success of the pregnancy, and the fetal placental unit is continuously exposed to microbial products coming from the maternal site. Yeah. This environment, this communication has led to what we call the equilibrium, the evolutionary equilibrium, that in order to survive, mammalian pregnancies have established a symbiotic relationship, first with the maternal microbiota, and second, with the maternal immune system. So in conclusion, pregnancy complications associated with immunological changes need to be evaluated with a different perspective. Each stage of pregnancy has a different immunologic status. So we cannot generalize that the whole pregnancy is an inflammation or is an anti-inflammation. And that is critical, especially if we want to treat or deal with women who suffer from autoimmune diseases. And autoimmune diseases exist, those associated with Th1 type of responses, like multiple sclerosis, rheumatoid arthritis, those are classical Th1, or Th2 autoimmune diseases like lupus. That is a disease that is affected by the production of autoantibodies that are attacking its own tissue. If a patient who has rheumatoid arthritis, for example, and she gets pregnancy, in the early pregnancy, the first trimester, when there is an inflammation, a Th1, her symptoms get worse. But then there is a shift into the Th2, and those patients do fantastic during the second trimester. There are some patients that have told me that, especially with MS and multiple sclerosis, that the best time of their lives is the second trimester of the pregnancy because they don't have the disease, it disappears. The disease comes back again at the end of the pregnancy when you have the inflammation. Again, just to give an example of the importance of understanding how pregnancy is a changing environment and it's a very complex immunological evolving condition. And the second one, which we are just trying, they're at the beginning of understanding what is going on, is the role of the microbiome. It plays a critical role, as I mentioned, by modulating the integrity of the reproductive mucosal. So it maintains the integrity of the mucosal in the vagina, in the uterus, and potentially even the placenta. And also has an important role on modulating the immune on regulating or promoting immune modulation. There is new data that we have now that the bacterial products enhance the production of immune modulatory factors such as interferon beta, PD-L1, Fas ligand, and so on, who are critical to prevent the immune rejection. So those factors enhance, in addition to the antigen presentation that we discussed before, the protection against the rejection, enhance the immune tolerance. But at the same time, they will enhance the awareness of the maternal immune system to respond quick and effective against the presence of viruses and pathogenic bacteria. In conclusion, the immunology of pregnancy is a complex biological process, and the last part that we don't know and needs to be elucidated is how those evolving immunological conditions have an impact on the development of the fetal immune system. Because an abnormal inflammatory response not